The human skeleton is well adapted for the functions it must perform. The organs of the skeletal system are bones, cartilage, ligaments, tendons, and joints. Functions of skeletal bones include support, protection, movement, mineral storage, and the formation of blood cells. Bones provide a rigid framework, collectively known as the skeleton, that supports and protects the soft organs of the body. The fused bones of the cranium surround the brain to make it less vulnerable to injury. Vertebrae surround and protect the spinal cord, and bones of the rib cage help protect the heart and lungs of the thorax. The skeleton supports the body against gravitational forces. The large bones of the lower limbs support the trunk when standing. Bones work together with muscles as simple mechanical lever systems to produce movement. In Week 2, we discussed inorganic salts. The extracellular matrix of bone is made of collagen and inorganic salts, such as the mineral compounds calcium phosphate, potassium chloride, magnesium sulfate, and sodium chloride and bicarbonate ions. Salt accounts for 70% of the extracellular matrix of bone by weight. Calcium and phosphate ions are the key components of bone minerals. They provide bones with structural support. Calcium is also required for our metabolic processes, including muscle cell contraction, nerve cell conduction, and blood clot formation. This is done to maintain an adequate supply for the metabolic needs of the body. When blood calcium levels are increased, different hormonal signals stimulate the storage of excess calcium in the bone matrix. The dynamic process of releasing and storing calcium goes on almost continuously. Think about the maintenance of homeostasis. For inorganic salt storage, also known as calcium deposition in bones, the bony matrix includes collagen and crystallized mineral salts known as hydroxyapatite. Hydroxyapatite is a type of calcium phosphate, an inorganic salt. Calcium is vital for balancing many of our metabolic processes within the skeletal system. For instance, the blood calcium level is regulated by two control centers that are endocrine glands. The thyroid gland regulates calcitonin, a hormone that promotes osteoblasts to deposit calcium. The parathyroid gland regulates the release of hormones that stimulate osteoclasts to break down bone to release the calcium deposits to balance the lack of calcium in the blood. For example, when our blood calcium levels are low, the parathyroid gland activates osteoblasts to break down bone tissue that results in more calcium salts in the blood. Alternatively, when our calcium levels are low, regulating osteoclasts reduce activity, and the hormone calcitonin activates osteoclasts to deposit excess calcium and store it in the extracellular matrix. In summary, endocrine gland mechanisms work with the skeletal system bone cells to regulate bone calcium deposition in a homeostatic loop. Almost all blood cells are formed in the red bone marrow of bone. In fact, 95% of our body's blood cells are formed in red marrow. Bone marrow is located within the spongy tissue inside most bones. Notably, all blood cells originate from stem cells. Stem cells differentiate and go on to form 95% of our body's blood cells within the red marrow of bone. As mentioned, all blood cells derive from stem cells. They specifically derive from the multipotential hematopoietic stem cell. Stem cells' proliferation, renewal, and differentiation are based on hormones such as cytokines and growth factors. For instance, a growth stem cell factor and protein enzyme that is such as thrombopoietin, TPO, is important for development and renewal of hematopoietic stem cells. Different enzymes will support cell differentiation and proliferation. What is hematopoiesis? The formation of blood cells is a process known as hematopoiesis. Hematopoiesis includes the development of erythrocytes, leukocytes, and platelets. Erythrocytes are more commonly known as red blood cells, and leukocytes are also known as white blood cells. Active platelets are smaller components of blood. Their primary role is to cluster together to create a plug to stop the bleeding of damaged vessels. Then they form a blood clot to end blood loss. Like all cells of the body, bones are dependent on oxygen and give off waste products in metabolism. Bones contain active tissues that consume nutrients, require an active blood supply, and change shape or remodel in response to variations in mechanical stress, such as physical exercise. The process of removing and replacing bone tissue is known as bone remodeling or bone metabolism. This vital process maintains bone tissue throughout the lifespan. In review, hematopoiesis, the formation of blood cells, mostly takes place in the red marrow of the bones. Red marrow functions in the formation of red blood cells, white blood cells, and platelets. When and where does bone growth and bone formation start? In infants, red marrow is found in most of the bone cavities. As we age and bones mature, the growth process slows. With age, red marrow is largely replaced by yellow marrow for fat storage. In adults, red marrow is limited to the spongy bone in the skull, ribs, sternum, clavicles, vertebrae, and pelvis. Before we explore the bone growth and repair processes, let's review the parts of long bone. Understanding the parts of bone will help with localizing the process of blood formation and regions of bone growth. The epiphyseal plate, spongy bone, compact bone, the medullary cavity, yellow marrow, periosteum, endosteum, and blood vessels are all parts of long bone. Let's further review the interior parts of long bone. The major portions of long bone are the epiphysis, diaphysis, and metaphysis. The epiphysis is located at both the proximal and distal ends of long bone. The diaphysis is the shaft of the bone. The metaphysis is the area between the epiphysis and diaphysis, where the shaft of the bone widens. Zooming into the inside of long bone, it contains many important features to the process of growth, repair, and connective fluids. The epiphysis forms a joint with another bone. The bone is enclosed by periosteum that is a vascular covering of dense connective tissue. The wall of the diaphysis is an area of cartilage that remains between the epiphysis and diaphysis. This is the epiphyseal plate or growth plate. Let's compare the compact and spongy tissue of bone. The names imply that the two types differ in density or how tightly the tissue is packed together. The names are also indicative of the relative difference in appearance, with spongy bone being more porous than compact bone. Compact bone consists of closely packed osteons or haversian systems. The osteon is the fundamental unit of compact bone. Let's review the structure of bone cells, a tissue, and the three types of cells that contribute to bone homeostasis. Osteocytes are star-like cells with many projections, known as cytoplasmic processes, that move towards the lacunae to the haversian canal through the bony matrix. The osteon consists of a central canal, which is surrounded by concentric rings, the lamellae of the matrix. The lamellae of matrix are characterized by the repeating pattern of rings within rings or concentric rings. Osteoblasts are bone-forming cells, activated by parathyroid gland hormones. Osteoclasts resorb or break down bone tissue, releasing calcium into the blood, that are activated by the thyroid gland hormone, calcitonin. Osteocytes are mature bone cells. These former osteoblasts have undergone ossification and are the direct result of this process. Osteocytes, formerly osteoblasts, are now surrounded by bone matrix. An equilibrium between osteoblast and osteoclast activity maintains bone tissue. There are two areas of bone tissue where calcium is stored. Compact bones store calcium in their concentric bony matrix, lamellae. Most calcium reserves are stored in spongy bone, a lattice-like structure of bony tissue. In review, compact bone consists of closely packed osteons or haversian systems. The osteon is the fundamental unit of compact bone. The osteon consists of a central canal, which is surrounded by concentric rings, lamellae, of matrix. Spongy bone is found in the interior of bones, and it filled with spaces and columns. Compact bone is found on the exterior of bone. Spongy bone is lighter and less dense than compact bone. Spongy bone consists of connected projections called trabeculae and irregular cavities that contain red bone marrow. The canaliculae connect to the adjacent lacuna cavities instead of a central haversian canal to receive their blood supply, exchange nutrients, waste, and signals throughout bone matrix. It may appear that the trabeculae are arranged in a haphazard manner, but they are organized to provide maximum strength, similar to braces that are used to support a building. The trabeculae of spongy bone follow the lines of stress and can realign if the direction of stress changes. In compact bone, the haversian systems are packed tightly together to form what appears to be a solid mass. The central canals contain blood vessels that are parallel to the long axis of the bone. These blood vessels interconnect by way of perforating canals with vessels on the surface of the bone. Compact bone consists of closely packed osteons or haversian systems. The osteon is the fundamental unit of compact bone. The osteon consists of a central canal, which is surrounded by concentric rings, lamellae, of matrix. Between the rings of matrix, the bone cells, osteocytes, are located in spaces called lacunae. Small channels, canaliculae, radiate from the lacuna to the osteonic, haversian canal to provide passageways through the hard matrix. Take a moment to distinguish between the two types of bone. The region labeled 1 is compact bone. It is much more densely packed than region 2, which is spongy bone. Let's apply our knowledge while observing micrographs of bone tissue. The micrograph samples we will review are tissue and cell samples of osteocytes, trabecula, endochondral ossification, the synovial membrane, bone marrow, and the epiphyseal growth plate. Micrographs are images captured through a microscope. The microscope enhances and magnifies slide sample details that would otherwise not be viewable with the naked eye. Let's view the microscopic details of bone to gain a better understanding of the parts that work together to complete the various functions of bones. A bone cell is an osteocyte. Here on this slide is a microscopic view of osteocytes. Let's take a closer look. The next few images were magnified with a microscope. Observe and compare the tissue and cell structures in micrographs that support bone remodeling and growth, contributing to skeletal homeostasis. In micrograph 1, we can see an elongated cell body with thin processes that stretch to the surface of trabecular bone. These are mature osteocyte cells. Osteoblasts migrate to the membranes and deposit bony matrix around themselves. When the osteoblasts are surrounded by matrix, they are called osteocytes. In this micrograph, mature osteocytes, formerly immature osteoblasts, are now surrounded by bone matrix. An equilibrium between osteoblast and osteoclast activity maintains bone tissue. This is a 400 times magnification of bone marrow tissue. The larger white circles are adipocytes. Adipocytes, BMAs, store energy in fat reserves, regulate insulin sensitivity, and regulate energy metabolism, secretes lipids or fatty acids to fuel osteoblasts and promote hematopoiesis. They also have an influence on osteogenesis. The hematopoietic stem cells are also known as the blood stem cells of bone marrow. They are the smaller purple cells in this micrograph. They are immature cells that will develop into various types of blood cells, including red blood cells, white blood cells, and platelets. These cells are tightly regulated to control the steady maintenance and balanced levels of blood cells. The larger purple clusters are megakaryocytes. MKs are large cells in the bone marrow that produce platelets that are essential for blood clotting. Let's look at mixed bone trabecula during endochondral ossification under the microscope. Endochondral ossification involves the replacement of hyaline cartilage templates with bony tissue. Most of the bones of the skeleton are formed in this manner. These bones are called endochondral bones. In micrograph 3, we can see a mixed bone trabecula. this tissue sample of mixed bone trabeculae, revealing a bluish center of acellular calcified cartilage. The bluish center of acellular calcified cartilage is covered by pink primary bone tissue containing osteocytes. Bone marrow is located among trabeculae. Mesenchyme is located on the surface of developing bone structures. It condenses into differentiated osteoblasts that later form the periosteum. The spongy trabeculae have disorganized-looking networks of blood vessels that supply the developing spongy bone with nutrients. In the spaces between trabecula is the red marrow where blood is formed. Osteoblasts mature to become osteocytes when this bony matrix completely envelopes the bone cells. After spongy bone is formed in the diaphysis, osteoclasts break down the newly formed bone to remodel the medullary cavity. This changes the perichondrium into periosteum. The osteoblasts form a collar of compact bone around the diaphysis. At the same time, the cartilage in the center of the diaphysis begins to disintegrate. Osteoblasts penetrate the disintegrating cartilage and replace it with spongy bone. This forms a primary ossification center. Ossification continues from this center toward the ends of the bones. As the cartilage is broken down by osteoclasts, the perichondrium changes into periosteum. The perichondrium and periosteum are distinct tissues that produce regulatory factors essential to long bone formation. Periosteum is a dense fibrous tissue that covers long bone. The outer layer contains fibroblasts that house the blood vessels and sensory nerves. The inner layer of the periosteum contains osteoblasts. The osteoblasts form a collar of compact bone around the diaphysis, middle region. At the same time, the cartilage in the center of the diaphysis begins to disintegrate. Osteoblasts penetrate the disintegrating cartilage and replace it with spongy bone. This forms a primary ossification center. Ossification continues from this center toward the ends of the bones. After spongy bone is formed in the diaphysis, osteoclasts break down the newly formed bone to remodel the medullary cavity. The epiphyseal growth plate of a developing long bone with the following layers can be seen in micrograph 4, a 100 times magnification of developing bone, ordered from right to left. Layer 1 is the resting hyaline cartilage zone. This layer is closest to the end of the epiphysis. Layer 2 is the zone of proliferating cartilage. This secondary layer consists of rows of cells undergoing mitosis. Layer 3 is the zone of hypertrophy. These are older cells that enlarge and thicken the epiphyseal plate. Layer 4 is the zone of calcified cartilage. This layer is composed of dead cartilage cells and calcified extracellular matrix. Layer 5 is the ossified bone of the diaphysis. When secondary ossification is complete, the hyaline cartilage is totally replaced by bone except in two areas. A region of hyaline cartilage remains over the surface of the epiphysis, referred to as the articular cartilage. An area of cartilage remains between the epiphysis and diaphysis, is the epiphyseal plate. In the fetal stage, future bones are first formed as hyaline cartilage models. Parts of the skeleton form during the first few weeks of life following conception. During the third month following conception, the perichondrium that surrounds the hyaline cartilage templates becomes infiltrated with migrating blood vessels and osteoblasts. This changes the perichondrium into the periosteum. The terms osteogenesis and ossification are often used synonymously to indicate the process of bone formation. Bones are very dynamic. The development and remodeling of bones continues throughout adulthood. As we age, bone development slows but continues to repair fractures and remodel bone to maintain the structure of bone in optimal conditions. The skeletal system begins to develop during prenatal development and continues to grow into adulthood. Bones begin to grow during the first few weeks of prenatal development. The formation of bones is known as ossification. There are two types of ossification: intramembranous and endochondral. After 8 weeks, the early skeletal template is formed in cartilage and connective tissue membranes and ossification begins. Bones form when bone tissue replaces connective tissue in one of two ways. Intramembranous bones are sheet-like layers of connective tissue. They are temporary structures of immature bone. Endochondral bones begin as masses of hyaline cartilage. As these bones mature, the cartilage is replaced by bone tissue. Let's further distinguish the two types of ossification. Intramembranous ossification involves the replacement of sheet-like connective tissue membranes known as mesenchyme with osseous bone. Intramembranous bones include certain flat bones of the skull, such as fontanelles, the sternum, and some irregular facial bones. During early development, intramembranous bones are first formed as mesenchyme, an immature connective tissue membrane. As they mature through osteogenesis, the bone-forming cells, osteoblasts, differentiate the embryonic connective tissue, replacing cartilage with bone. Osteoblasts migrate to the membranes and deposit bony matrix around themselves, until they are surrounded by the bony matrix called lacuna. Once osteoblasts are enveloped by lacuna, they are considered mature bone cells called osteocytes. Most of the bones of the skeleton are formed through the process of endochondral ossification. Most of the bones of the skeleton are formed in this manner. Bone created by the replacement of hyaline cartilage templates with bone tissue are called endochondral bones. In this process, the future bones are first formed as hyaline cartilage. As the cartilage is broken down by osteoclasts, the periosteum develops. Periosteum blood vessels and osteoblasts differentiate and invade the disintegrating cartilage as the osteoblasts beneath the periosteum start depositing compact bone. Osteoblasts become mature osteocytes once the bony matrix surrounds them. Bones grow in length at the epiphyseal plate by a process that is similar to endochondral ossification. The cartilage in the region of the epiphyseal plate next to the epiphysis continues to grow by mitosis. The chondrocytes in the region next to the diaphysis age and degenerate. Osteoblasts move in and ossify the matrix to form bone. This process continues throughout childhood and the adolescent years until the cartilage growth slows and finally stops. Essentially, the growth plate thickens and shifts towards the articular end of the bone. Simultaneously, old cartilage is calcified and ossified on the side of the plate adjacent to the diaphysis. When cartilage growth ceases, usually in the early 20s, the epiphyseal plate completely ossifies so that only a thin epiphyseal line remains, and the bones can no longer grow in length. Now that we have a general overview of bone growth and development processes, let's examine some of the homeostatic disruptions of the skeletal system. Osteoporosis is a condition developed when there is a reduction in the amount of bone mass. With osteoporosis, the balance between calcium deposition and calcium loss in bones is disrupted. Thus, the bones lose more calcium than they deposit. As a result, the bones become brittle and break easily. Cortical bone becomes more porous and thinner, while the structure of trabecular bone is impaired. This makes the bone weaker and more likely to fracture. Osteoporosis is most common in postmenopausal women because of the hormonal changes that happen at menopause directly affect bone density. To the left is healthy trabecula, to the right is thinning and brittle trabecula. For example, the female hormone estrogen is essential for healthy bones. After menopause, estrogen levels fall. This can lead to a rapid decrease in bone density. The lack of vitamin D and calcium can lead to osteopenia in adults and rickets in children. This condition is the softening and deformation of bone due to lack of essential vitamins, minerals, and nutrients. Vitamin D and calcium. Scoliosis is the curvature of the spine that can occur during the growth spurt ahead of puberty. In 80% of cases, the cause of scoliosis is not known. Most cases are mild, but scoliosis can be more common in those that have pre-existing conditions such as cerebral palsy and muscular dystrophy. The other 20% of cases can be the result of degeneration of spinal discs from arthritis, complications from osteoporosis, or a hereditary condition. The micrograph to the left depicts the atypical cells of bone marrow cancer. also referred to as multiple myeloma. This cancer starts in the bone marrow and can spread to other bones. This condition is the formation of atypical cells in a group of plasma cells, crowd healthy cells becoming cancerous, then multiply causing damage to the bones, as well as other interconnected organ systems. To the right is a micrograph of a bone tumor. This is the development of a tumor in the bone. There are 6 types of fractures that can occur with bone injury, including greenstick, fissured, comminuted, transverse, oblique, and spiral. We will view these in detail in this week's lab. In summary, the human skeleton is well adapted for the functions it must perform. The functions of bones include support, protection, movement, mineral storage, and formation of blood cells. There are 2 types of bone tissue: compact and spongy. Compact bone consists of closely packed osteons or Haversian system. Spongy bone consists of plates of bone, called trabeculae, around irregular spaces that contain red bone marrow. Osteogenesis is the process of bone formation. 3 types of cells, osteoblasts, osteocytes, and osteoclasts, are involved in bone formation and remodeling. In intramembranous ossification, connective tissue membranes are replaced by bone. This process occurs in the flat bones of the skull. In endochondral ossification, bone tissue replaces hyaline cartilage models. Most bones are formed in this manner. Bones grow in length at the epiphyseal plate between the diaphysis and the epiphysis. When the epiphyseal plate completely ossifies, bones no longer increase in length.